Hi, Bio 161 students. So this week we are talking about cellular respiration. And in order to get a better look at this process, we need some sort of indicator that tells us whether or not cellular respiration is happening and to what extent it's happening. An indicator is a substance that gives us some sort of measurable effect for a process that is occurring. So to measure cellular respiration, we use cabbage extract as our indicator. Cabbage extract is a really interesting substance because it changes color when it changes pH. So right now it's this blue color, but if I were to make it more acidic, it would turn more purple or pink in color. And the reason we can use this to measure cellular respiration is because CO2 is a byproduct of the reaction. So if I added CO2 to this cabbage extract, it'll lower the pH of the cabbage extract and make it change color. Importantly, note that ATP is the goal of the cellular respiration reaction, and the point is to make ATP for the cells to have energy, but ATP is a lot harder to measure. So since CO2 is a byproduct of this reaction, we can measure that instead. And since if CO2 is being made, we can assume ATP is also being made, and it gives us a sense of how much cellular respiration is happening. So I have some cabbage extract here, and I have some vinegar, which is acidic. You would be bubbling CO2 into the cabbage extract, but I'm just using another acidic solution just to show you guys what that color change looks like. So I'm gonna put some vinegar into this glass, and then I'm going to add my cabbage extract. So you can see that it has turned this really light pink/purple color, and that's because now we have made the solution more acidic and it's changed color. So this tells us that cellular respiration, in quotes there, has occurred, but how do we know just how much cellular respiration has occurred? Well, the cool thing is that this reaction is actually reversible. So if I add a base to this solution now, it'll turn back to blue. In the lab, we'd use drops of ammonia, which is a basic solution, to bring the solution back to blue, and the amount of drops of ammonia that are needed to change the color back to blue is proportional to how much cellular respiration has occurred. So the more cellular respiration, the more CO2 bubbled into the cabbage extract, the more drops of ammonia it would take to make the mixture blue again. So to show you that reaction, I have another basic substance here, just sodium bicarb. Remember that you would use ammonia drops to do this in the lab, but this is what I had in my house. So I just want to show you guys what that color change looks like. So if I add some of this to my mixture, it's gonna bubble and spill all over my desk because that's what bicarb does. But if we let it do its thing for a little bit, we can see that you can kind of see that it's turned back to blue. And that is pretty much the color we started with. So you can see the solution's blue again, and that is how we measure cellular respiration in this lab, using cabbage extract as an indicator for CO2, one of the byproducts of cellular respiration.